Welcome, everyone, and good evening. Thank you so much for joining. We are so excited about tonight's program. This is a huge time in the CF community. For the first time, we have various gene-based therapy trials that are open and more coming soon. And I think I probably speak for many of you when I say it's about time, thank goodness. But with that progress comes a lot of questions. And so the purpose of tonight's town hall is to share information about gene-based therapies and to really, the biggest purpose of all is to answer your questions. We want to bring the community together and hear what's on your minds, hear what you're excited about, hear what you're worried about, and provide as much information as we possibly can. And so tonight, what we're planning to talk about is gene-based therapies in general, what to expect with gene-based therapy trials, and what to consider and to ask when you're thinking about participating in a gene-based therapy trial. So the game plan for tonight is we're going to start with a brief overview of gene-based therapies from an esteemed group of CF clinicians and a person with CF, and I'll introduce them all shortly. Then we're really going to spend the vast majority of tonight answering your questions, the questions that were pre-submitted when you registered for this event, and then also we're going to open it up for a live question and answer session. So our goal, as I said, is to answer what you are wondering about and what you care about. So please tell us what's on your mind and don't be shy. That's why we're here. But first, before we get into all of that, I want to start by introducing myself and emilys-entourage for those of you that are not familiar. I am Emily Kramer-Golinkoff. I'm a co-founder of emilys-entourage. And emilys-entourage is a 501c3 that speeds research and therapeutic development for those in the final 10% of the CF community that don't benefit from existing CFTR modulators. I'm also a person with advanced stage CF myself. We started emilys-entourage back in 2011 to speed life-saving advances for those in the final 10%. And at emilys-entourage, we believe that every single person with CF deserves to live a long and healthy life, no matter how rare your mutation. And so we race every single day to make that dream a reality and to make it happen fast, because we realize that time matters. So we've come a very long way since those early days in 2011. Since then, we've raised over $11 million. We've awarded 32 research grants to top academic institutions around the world, and we secured over $42.8 million in follow-on funding. We've advanced 3 research projects to clinical trials and multiple others to the preclinical stage, including Spirovant, the now acquired CF gene therapy company that EE launched through venture philanthropy. We also developed and we maintain a CF nonsense mutation patient registry and clinical trial matchmaking program to expedite clinical trial recruitment and to inform the CF community directly about clinical trial opportunities. And really, all of our work is singularly focused on just one thing, and that is accelerating research and drug development for people with CF who don't benefit from existing modulators. But tonight is all about gene-based therapies. We are so lucky to be joined by a panel of brilliant and compassionate CF clinicians and an amazing person with CF. We're deeply grateful for their time and devotion to the CF community always and in joining us tonight. And so with that, I'd like to ask them to briefly introduce themselves, and starting with Dr. Marty Solomon.
Hi, everyone. Marty Solomon. I'm an associate professor of medicine at the University of Alabama at Birmingham and direct the adult portion of our Therapeutics Development Network Center and have been involved with CF research for more than almost 15 years now. So it's really my pleasure, and we look forward to tonight and having a good dialogue with you.
Hi, everyone. It's Greg Sawicki. I'm a pediatric pulmonologist and the CF Center Director at Boston Children's Hospital. And like Marty, I've been involved in CF research for almost 20 years.
And I'm Jen Taylor-Cousar. I'm an adult and pediatric pulmonologist at National Jewish Health. I run our Therapeutics Development Center and co-run our center. I also have been involved about 20 years, and I'm really happy to be here and happy that emilys-entourage is bringing this out to the community.
And hello, everybody. I'm Ahmet Uluer. I'm a medicine and pediatric trained pulmonologist like Jen, and director of the adult CF program at Boston Children's and Brigham and Women's Hospital here in Boston. And also been doing this for a long time. And I'm grateful to emilys-entourage and to everyone here for the invitation to speak with you today.
Hi, my name is Laura Mentch. I'm a person with cystic fibrosis. I live in Bozeman, Montana, and I've been engaged with the CF community for the last 20 years, just like our presenters.
Thank you all so much for those introductions. So before we dive into the presentation tonight, I just want to share a few housekeeping items. So the first is that the program is being recorded. So all participants are off camera, but the question and answers will be captured to continue to share the information beyond tonight and to inform future discussions. So closed captioning has been turned on for tonight's event, so feel free to activate it on your own account if you'd like to see the captions. If anyone has a question that they'd like to submit later tonight, and we hope that you do, to do so, you enter the question into the Q&A box and then just click send, and then we will receive all of those on the back end. We will do all our very best to answer as many questions, but we're not going to be able to get to all just by the sheer volume of questions we've received. So we're going to start with a group of pre-submitted questions, and we're going to ask you to hold your live questions until we open it up for the live Q&A session. And any questions that we're not able to get to in either the pre-submitted section or in the live section, we are going to aim to put out a blog post recap that hits on some of those questions afterwards. So keep your eyes peeled for that in the weeks to come. I also want to just make a note that we're not able to answer any personal medical questions tonight. So if you have any questions pertaining to your personal medical situation, we encourage you to talk with your own healthcare team about those as well. Finally, we have a very brief survey at the end of the event, and we really want to encourage you to do it. It takes less than 60 seconds, but your feedback is incredibly helpful. So when you leave the event, I think it'll pop right up, and if you could just take a few seconds to do that, we'd be so grateful. And actually, finally, I have a very brief legal word. So the speakers, as well as emilys-entourage, may have financial relationships with commercial interests, including support for campaigns, events, recruitment, and programs. And you'll see more about this on forthcoming slides. These relationships do not affect our opinions shared this evening or signify any endorsement of specific trials, drugs, or companies. And I also want to make special note that tonight's event is not supported by any outside companies. So thank you so much again for being here. And without further ado, I will pass it on to Dr. Sawicki to get us started on the overview presentation on gene-based therapies.
Thanks, Emily, and thanks again for inviting all of us to present. And as you did mention, there are a few disclosures that all of us have as physicians and researchers in the field of CF, and we'll just scroll through them. They're available, but I would just say there's 2 slides of disclosures, and it's just a testament to how the 4 of us, but also hundreds and hundreds of other CF clinicians and researchers are working tirelessly with various companies in industry and in academia to try to advance the field. And so what we're going to do tonight is really do an overview of the genetics of cystic fibrosis and carry that through to how modulator therapy works, and then how that differs from some of the approaches that are being tried and studied in terms of gene-based therapies. And so we'll go back to the basics to start. And those of you on the webinar who have CF or who live with people with CF or are involved in CF care or research in some way know that CF is an inherited disease. And so fundamentally, it's caused by inheriting mutations or variations in the gene that are inherited both from a mother and a father. And so in order to have cystic fibrosis, we are aware that an individual with cystic fibrosis has to have 2 genes that both have a variation or mutation. Next slide. But fundamentally, what genes do in our body is they create proteins. So our DNA makes up code for millions and millions of proteins in our body. And in 1989, when the gene for CF was identified, it was known then to code for a protein called CFTR. And CFTR is essentially the protein that is in the epithelial or lining cells of the body, throughout the body, that when you have errors or mutations or variations in the genetic code, that protein doesn't function normally. But there are lots of different ways that it takes for DNA to then lead to protein. And so we'll go back to biology 101 to really talk about these different steps. And that's important because when we think about gene-based therapies, the different approaches are tackling different parts of this pathway. So we start in every cell that has a nucleus, that's where your DNA lives. The first step is for DNA to be transcribed in a process called transcription, made into something called messenger RNA, or mRNA for short. mRNA, you'll hear about later on, became famous during the COVID pandemic for the vaccine strategies, but is really the sort of next step from DNA to protein that the cells use. This mRNA then undergoes a process called translation. And in that point, the mRNA code is then turned into a protein code. And each of those little letters that are in a genetic code identify a specific amino acid. And if you remember your biology, amino acids then turn into proteins. And so you have these two processes that go from DNA to mRNA to protein. But then the protein is usually inside a cell and has to get to where it needs to go. And in the case of CFTR, it's a complicated process where it has to go through parts of the cell to make it to the surface. And ultimately, there's modification that has to happen to make it to the cell surface. And ultimately, if you don't have any problems with the code, that protein makes it to the surface. But as we know, there are various different ways that CFTR can be mutated to lead to dysfunction of the protein. And I think with that, I turn it over to Marty.
Great. Thanks, Greg, for getting us started. So one way that we want to introduce these concepts to you all is to simplify this a little bit from the basic biology into a set of analogies that can help you understand the interactions between protein modulators and the DNA to protein process, because understanding those two things will help us understand gene therapies a little bit better, so we can think about how we design and conduct studies to test these type of therapies. So in the analogies we're going to introduce the next few slides, we're going to think of DNA as a hard copy, think about in your computer, of a set of genetic instructions, or a hard copy blueprint for building proteins. The mRNA that Greg just talked about is a photocopy or a portable blueprint copy to take to the site where the proteins would be built. Like, for instance, if a builder takes a set of blueprints onto a building site to construct something. The ribosome is a portion of the cell that uses the blueprint instructions to assemble the building blocks to build a protein. And the protein is really, in this case, the ribosome can be thought of as the builder. And the protein is really the end product of what the builder has built. So a machine or a structure that performs a function in the body. In the case of the analogies we're going to learn about in the next few minutes, that protein is the CFTR protein. It's going to be represented as a gate, because that's what it does at the cell surface, is it functions as a gate to help us regulate the movement of chloride and other anions, which are just ions that float around in our cells and inside and outside of our cells. In the case of CF, the disease happens because of the absence of the protein function, the CFTR protein function, which derives from an issue or a misprint or an error in the hard drive copy in the genetic material all the way back at the first of this process. And in the case of our analogy, it'll be absence of a gate to move those ions to control and regulate the movement of mucus in our body, especially in the lungs and the organs where that's very important. So this is the kind of rubric for understanding the next set of analogies. So let's think about our blueprints and gates in relation to the types of mutations. All of you are probably familiar with the types of mutations that you may have, and we have learned that from your clinical doctors. In the case of these mutations, different defects in that protein, the DNA to protein process are happening to cause CF disease. Class 1 mutations, which are mutations in which we call premature termination codon mutations, often designated with an X in the mutation type, are situations where the blueprint doesn't call for making gates at all. In other words, the protein is foreshortened and it does not therefore make a gate at all. Class 2 mutations are where the mutation causes the blueprint to only call for a few faulty gates to be made. An example of this is the F508del mutation, a very common mutation. And in the case of untreated F508del mutation, mutated CFTR, only a few poorly functioning gates will reach the cell surface to do their job. The case of class 3 and 4 mutations are various levels of dysfunctional or lack of functioning gates to be present on the cell surface. In the case of class 3 mutations, like the G551D mutation, which was the original mutation that ivacaftor was originally approved for in CF going back over a decade, this mutation results in a gate that's present at the cell surface, but it's locked shut, so it therefore can't do its job. The class 4 mutations work in a similar fashion in that the gate is present at the cell surface, but the gate is blocked. In other words, the tunnel through which and the opening through which the ions move is tortuous and difficult to get through. And finally, class 5 type mutations cause the blueprints only to make a few gates. So analogous to class 2 mutations, the number of gates made is inadequate to keep up with the need to not have CF-related protein problems. In the case of not having CF, there's an appropriate amount of gates that open normally on the cell surface. So let's turn now to an analogy that may help us to simplify and think about this further. In these analogies, we're going to think about the interactions between mutation types and treatments that we know that have been shown to be tried and true, like CFTR modulators. And in the case of these analogies, we have a set of cows who represent the ions that want to move, and they want to move from a barren pasture over to a very bright and sunny pasture to be able to eat grass. In the case of the G551D mutation, these cows cannot get through because the gate is locked shut. However, if we give a special key to the gate known as a potentiator, Kalydeco being one of those types of mutations, one of those types of drugs known as a CFTR modulator or potentiator, we can unlock the gates and our cows can get over to a happy green pasture. Now let's think about a more complicated mutation, the F508del mutation. It's very common in people living with CF. The F508del mutation, as you recall, the genetic blueprint defect or error calls for an ineffective number and ineffectively functioning gates in our fence, separating our cows from their green pasture. So if we take the F508del mutation and have our builder treated with a corrector like elexacaftor or lumacaftor or tezacaftor, some of which are the components of tricafta, and then we will get a few more gates to be produced in our fence, which represents our cell surface. However, in the case of F508del, the mutation causes the builder to have created some additional gates where they should be present, but they're not able to open fully. So if we then add a potentiator modulator like Kalydeco in combination with these corrector molecules, then this will allow the faulty gate to be opened up and our cows again to reach to the greener pastures as they should. And we have happy treated cows and a better functioning situation with cystic fibrosis. So now I'm going to turn it over to Jen, and she will talk to us about developing treatments and cures for all in this rubric with gene-based therapies.
Thanks, Marty. You go to the next slide.
Yes.
I think these are still your cows, but one of the things that is really important and is really relevant for this group is that there are some mutations that cause problems with the blueprint. So you have no gates at all. And because you have no gates, these mutation types, they can't be treated with modulators. Next slide. So with gene-based therapies, and sometimes you'll hear them referred to as nucleic acid therapies or genetic therapies, what we're trying to do is restore the message by replacing or fixing that blueprint so that there's a plan that actually builds the gates so that they can open. Next slide. You can keep going. Sorry, they're really animated. You can keep going. I'll go on to the next one. So that's the theory behind all of it. What I'm really going to focus on is thinking about how we test these in clinical trials. Okay, next slide. So currently, we use 4 strategies to treat CF, or we think about 4 strategies. So there's the traditional therapies like Dornase alpha or pulmozyme and hypertonic saline and enzymes. Those are really meant to treat the signs and symptoms that we see in people with CF. And Marty just explained to you that the CFTR modulators are really meant to fix the protein problems. But what we're really moving towards now is looking at RNA, that message, and replacing the RNA or replacing tiny bits of it with short nucleotides that I'll show you called ASOs in a minute. And then DNA, so replacing the gene, and that's with gene delivery. But ultimately, what we think we need to do is edit the gene, correct that specific mutation that somebody has. Okay, next slide. So again, and I'll say some of these things over you and some of the things that Marty said, because I think that it's really important that some of these messages get through. What we're trying to do with gene-based therapies is reprogram the cell to make a functional protein. So we're going to provide the correct instructions either with DNA or with RNA or parts of RNA. And that tells the cell, make this correct protein or the gates. And that's what our goal is. So next slide. So Marty used cows for an analogy, and I'm going to borrow the one that Dr. Martin Mentch and Hillary Valley used at the North American Conference. I feel like that was a couple of years ago. But if you think about CFTR being a car that needs to be repaired, like you really need to get where you're trying to go, but your car is not working. So with mRNA therapy, what we're going to do is something temporary. Next. And basically with mRNA therapy, it's like having a rental car because it's not going to last very long. And you're probably going to have to take it multiple times. Next. With DNA therapy, what we're going to do is replace the gene. So we're just going to replace it all together. It's like having a brand new car. And then finally, with gene editing, we're again going to go in and we're actually going to see, oh, that's what the problem is. We're going to fix that problem right there and solve it permanently. Okay, next slide. So as Greg mentioned, back in 1989, when the CF gene was discovered, we all thought, great, we'll have gene therapy around the corner because it's a single gene and we'll easily be able to fix it. And in fact, the studies started pretty soon after that, but there were problems. So one is they couldn't get enough of the corrected gene into the cells. They got it into the nasal cells, the ones in the nose, but they couldn't get it into the lungs. Also, they were using a virus to help transport it into the cells. Well, your body's natural response to a virus is to try to fight it off with infection. And also there's thick, sticky mucus in the lungs of people with CF, obviously. And so trying to get past all of that with the corrected gene turned out to be quite difficult and unfortunately did not work. So as a research community, we started thinking about, again, like, let's go back to the drawing board. We need to find a therapy for every single person with CF. So when we're thinking about it, even before you get into the trials, we're like, well, which virus do we use? Do we use a virus that's kind of small but has less side effects, or a bigger one that may have a few more side effects? Or do we use these little fatty particles called lipid nanoparticles, or LNPs? And then there are lots of different cells in the lungs. Which ones do we have to get the corrected gene or the RNA into? And how many of them? Do all of them have to be corrected? Or if you got 10 or 15%, would that be enough? And then the next question is what efficacy? In other words, what measures of how well it's working do we need to see in cells or in animal models before we say, oh yeah, let's try this in people? So many of you will be aware that the modulators were all tested in cells, and that turned out to be very closely correlated. It followed very closely what happened in the clinic. So we don't know what that is yet for gene therapy. And then once we go into the early phase studies, again, what do we measure to say that they're actually working? So with modulators, because it's given to your whole body by taking it orally, you can measure a sweat test, but we don't actually have a sweat test for the lungs. And then again, if we're using a virus or maybe even with that lipid nanoparticles, your body's reaction may be to fight it off with inflammation. And how do we measure the inflammation and how much is too much and is going to cause trouble? And then finally, with the clinical trials, again, what do we measure to say it's working? How much improvement is enough? So we saw with some more modestly effective modulators that even 2 to 4% improvement in lung function was enough to stabilize people and prevent them from having exacerbations. And then finally, which people with CF are included? Do we only give these therapies to people who don't have eligibility for modulators, or what about those people who are eligible but can't tolerate them? Okay, next slide. So those are the questions that researchers are asking. What the community has been asking us is, how will these trials look? So are all people with CF going to be eligible? What's the dosing schedule going to be like? How will we get it to people? And is it a permanent cure? So in this table that Dr. Solomon created, he has checks throughout. So for example, for mRNA, it'll be for all people. It's probably going to be weekly, maybe even daily. It'll be delivered to the lungs, and potential challenges include, we don't know which cells are okay to have it in. Is it okay to have it in cells that don't usually have it, or is that going to cause side effects? But it is not going to be a permanent cure. For gene delivery, again, all people should be eligible. We don't really know how, since we've never had a successful therapy, we don't know how often we'll need to give it. Maybe yearly, maybe every few years. Again, it's going to go first to the lungs. It'll be inhaled. We don't know, again, if you put it in cells that don't usually have it, does that cause a problem? And will antibodies cause a problem for re-delivery? It's not going to be a permanent cure. And then finally, gene editing, which is a little bit far away still. We don't know what the dosing schedule would be. We do know that we'll probably give it throughout the body. We're not sure if it's going to cause editing into other cells and we want it to cause editing in, but it would be a permanent cure if we can get it to work. Okay, next slide. There are also some really important considerations that we have for who is going to be included in these trials. So many of you are aware that the percentage of people who are Black, Indigenous, and people of color who are in that percent of people who don't have a modulator are overrepresented. There are more people of color in that group. Next, Marty. But we also know that we need to include people, we need to ask people who, regardless of their background, about participating in these trials, but it's important that the consent is very different than the consents that we've done, for example, for the modulators or for an inhaled antibiotic. Next. Our protocols are going to likely require very long and multiple days and sometimes inpatient stays with frequent in-person follow-up. And this is absolutely necessary because these therapies are things that we haven't tried before, and we have to make sure that they're safe. We do not want to advance these therapies so quickly and find out that we are causing people harm. So they seem really rigorous and onerous, but it's because these are very, very early trials. And most of these therapies, are the first time they're going to be given to somebody, if it's an actual person and not an animal or a cell, is going to be somebody with CF. So we have to be very, very careful. Next. We think that for our gene therapy, at least redosing is going to be necessary, but antibody development, again, your body wants to fight off viruses, may be a challenge for giving another dose. Next. Also, early phase study participation, particularly for gene therapy, probably less so for mRNA, might prevent you from being eligible for future studies, although not for future therapy. Next. And then the currently proposed approaches are going to improve your lungs, hopefully, but they're not going to affect anything else in your body. It's like a lung transplant where you replace the lungs, but the rest of your body would still have CF because these early therapies are just being delivered to the lungs. Next. So people asked about what do we know about these studies, which ones are soon to start, which ones are already started. And I'm not going to read all of these to you, but there are websites particular to the companies, but also you can go to clinicaltrials.gov and you can search cystic fibrosis and gene therapy, and it'll show you which trials have already started and which will be starting soon. And these are the ones that are in the US currently. Next slide. There are 2 that are currently still in the preclinical or science phase that should be in phase 1 studies soon. Next slide. And there are also some that are happening outside the US, both for restoration of CFTR, like fixing the protein, as well as for gene therapy. Next slide. And with that, I'll turn it over to Ahmet to talk a little bit more about how we review these protocols and think about the safety.
Wonderful. Thanks so much, Jen. And thank you again, everybody. So we want to talk a little bit about, Jen just spoke a little bit about what we want to measure to make sure gene therapy is working, and the outcome measures are going to be one of the challenges that we're facing. As she mentioned, it's not a systemic, not systemic at first. And so we're going to have to figure out how we're going to measure that in the long. And then we're going to think about enrollment. Jen, shared with us the challenges that we're going to have with long days, the time commitment and everything else that's going to come with, joining these clinical trials, but also the concern about safety. And so when looking at these challenges, I put safety, right up front here, because, gene therapy trials have been going on for a while. And I think a lot of memory goes back to the 90s, where there were, clinical trials being run, and we identified, serious health risks and toxicities, inflammation, and even cancer. So we've come a long way since then, and there are a lot of different, ways that we have changed the way that we're approaching these therapies. So hopefully I'm going to share with you some of the pathways in order to ensure the safety of those who are joining these clinical trials. And, and there are, lots of successes. There are a lot of other, chronic illnesses that have been benefiting from gene therapy. But of course, we're still in the, the early phase, and there's some unpredictable risks that are there. And that's the reason why, there are a lot of people out there, a lot of agencies that are looking out for your best interests. Next slide. So this is just a slide that's going to take us through the different ways that we're looking at safety in clinical trials before and during your study. And just next slide will highlight before enrollment. If you can just click forward, there we go. It first starts with the Food and Drug Administration. The Food and Drug Administration has actual guidelines. There's a cellular and gene therapy guidance that allows people, academia, sponsors who are looking to join the gene therapy clinical trials, guidance on how to go about it and what the FDA is looking for, with safety being right up there. So the study sponsor works with the FDA, very closely as they start thinking about this and taking what they've learned at the benchtop or in the lab, bringing it to animals and then to humans. And that's the study sponsor who's bringing that idea to the FDA. And in CF, we have this therapeutic development network. You've heard of it. It's an incredible organization of CF centers across the country working together to, not just, monitor safety, but to make studies efficacious and, easier to complete and get to you and the consumer, just like the modulators. And we couldn't do it without the whole community helping. And the Therapeutic Development Network is that scientific review that makes sure, and we're going to go through that pathway just in a minute, but make sure that we've really checked all the boxes that we want to make sure that this is as safe and efficacious as it can be. And then there's a data safety monitoring board, a group of experts who are monitoring safety before as they study the protocol, and then during the study. And then the IRB, there are central and local IRBs. These are institutions or review boards locally at the institution that you're being cared for, or centrally for trials that are being done in multiple institutions, making sure that there's another level of oversight and safety. And then your site study team, we also review each study and making sure that we feel it's safe and that it really is the best study for our CF community. And then, of course, the patient's role and your role is to make sure that you know everything there is to know about the study, and that you're getting consent, and you're informed, and that you are, that you're going into this with the appropriate knowledge. The next slide will just highlight the Therapeutic Development Network and a scientific review process, and we'll make sure. And then also wanted to just cite that with Emily's Entourage clinical trial matchmaking program, The clinical review committee is, they have an incredible group of clinicians and researchers and individuals, CF, reviewing protocols as well and making sure that the goals and mission, are safety and making sure that the clinical trials are, in your best interest. And so, emilys-entourage has been, looking out for those people who are not eligible for modulators and really spearheading, the next stage of incredible, the studies that are being done. And one such example is one of the sponsors is Spirovan, looking at lentivirus, but also many others. So, but the next slide, or maybe two more, there we go, the Therapeutic Development Network. So this pathway, I happen to be one of the people working there with the TDN. And when we're, and next slide, it first starts with the sponsor, talking to the FDA, but also talking to the CF Foundation very early on and to the Therapeutic Development Network. And there is a group called the Executive Committee, the CREC, that looks at this at a very high level. And there's a lot of discussions taking place with the sponsor before we even get to the next step, which is, and next slide, the protocol review. And for many studies that would be, where things, the protocol review board just, which is comprised of clinicians, research assistants, research nurses, statisticians, but also people from the CF community to review a protocol. But in addition, with the clinical trials that are gene-based therapies, we have the gene therapy working group, and that is headed by Deepika Polanani and Dr. Manu Jain, who are also experts in this field, and they will help identify any concerns. And then we go back and forth talking about it amongst ourselves, and then we send a decision, next slide, to the sponsor, and we will let the sponsor know often that what issues were raised. And I will tell you that the CF community member and their feedback is weighted very heavily, and as with everybody else, in the scientific merit of the study. And thus that goes back to the sponsor, issues are identified, and then often the study is deferred for further discussion and edits. And next slide. And then it goes back to communication to the CF community and your centers, where we talk about, because we all hear about these studies that are in the pathway, and we just are keeping each other up to date. And then next slide. Then we are going back to the protocol review in the gene therapy working group. Next slide. And then ultimately, the study and the protocol gets sanctioned by the TDN, and that gets communicated. And next slide. And this process is just really a back and forth process that happens, but it takes a really long time. And you may be wondering, about why things take so long, but it's really because of safety, because we want to make sure that study is feasible. If you're being asked to be in the hospital for weeks on end and get multiple blood draws, that may not work out, and everyone is working and has other things going on. So we need to make sure that not only they're safe, but they're also feasible to you, and that also there's scientific merit. In next slide. And so the next part is during the study. So it just doesn't end there when the clinical trial starts and you've signed informed consent. The FDA is, next slide, constantly looking at review of the study to make sure that it continues to perform in a safe manner. The study sponsor and the CROs that they hire are looking to make sure that the adverse events are being recorded and that they're being thoroughly investigated and explained. The Therapeutic Development Network continues to provide study oversight, and they rely also on the Data Safety Monitoring Board to make sure that they're keeping an eye on the study, and that the Data Safety Monitoring Board is always expecting to learn about adverse events and to make sure that they're giving the proper feedback. The IRB is doing the same thing locally and centrally. They're also responsible to evaluate adverse events and anything that might be an unintended part of the study. And then, of course, your site study team is doing the same thing. And your role is to make sure, as a patient who's involved in the study, to report any adverse events and to keep everybody informed in order that, we're making sure that the study is going safely, and that we're identifying things early on before we move forward. Next slide. So I'm gonna hand things over to Emily, and thank you so much for this opportunity again, and we will be answering some questions.
So thank you all so much for that great overview. I love the cow and the car metaphors. I just want to remind everyone that if you have a question, please submit it in the Q&A box, and that allows us to sort of look at all the questions and hit as many of them as we can. So, the physician perspective on gene-based therapies and clinical trials is invaluable, but as we all know, the experience of actually being in a trial is quite a different thing. And so that's why we are so thrilled to be joined tonight by a person with CF who has participated in a gene-based therapy trial in the past. And so with that, I'm so pleased to welcome Laura Mentch. And Laura, I'm wondering if you can share a few words with us about your experience as a past trial participant. And Laura, I think you're actually unmuted.
I am muted. Now I'm unmuted.
Perfect.
Thank you,
I
Laura.
want to start back to, about 20 years ago, I entered CF care with my late diagnosis. And my very first clinic visit was long visit. And they gave me all these medications, machines, and treatments that I'd never had before. And it was a lot. And it was also new. The last med I'd taken prescribed by my allergist was theophylline. But I was grateful to have someone who finally knew how to take care of me, but overwhelmed with the task and the costs of CF care. And I did not stop to wonder what it took to make all these medications available to our small community and to me. While starting to organize my days and treatments, my days around all the treatments, one of the nurse practitioners at the clinic where I worked let me know that her sister was on the staff at the Center for Biofilm Engineering at Montana State. It's just a few blocks from my home. She told me that her sister believed that the key to CF would be found in gene therapy. But at that time, so many years ago, that went over my head. I did not even know what a biofilm was. And we have this very famous institute right here. But I did begin to hear about clinical trials, and I asked the nurse coordinator about them. And she was discouraging because of the cost of travel for the many visits most studies require, and that I live a very long drive or plane flight from my CF center. When support for travel was included by study sponsors, it became practical for me to participate. I learned about clinical research studies from many places, and we've got with online so much information, conferences, presentations, emails, newsletters, my CF peers, the clinical study finder, and now I've learned about the matchmaking program at emilys-entourage if you have a nonsense mutation. So the CF team will also maybe invite me into a study before I've become aware of it. And before I do anything about screening, I sit down with the nurse coordinator and my CF doc. Do they think this is a good study for me? If they give me the green light, then I feel more comfortable going forward. I really do trust their guidance for me. But when I'm entering a study, still a couple of things I need to think about. Am I eligible for the study? Do I meet the criteria? What about the day of screening when my labs or FEV1 cooperate so I can be in the study? Do I have the time to give to the schedule of the study, or does the study fit into my schedule? And to process any concerns I might have. And do I want to do this study at this time? I do find it's always has been an opportunity. I haven't declined a study yet, and I look forward to participating more in the future. But being part of research studies teaches me new things. I've had some tests where they were old tests, but new to me. I had the lung clearance index and the nasal potential difference. So it's been, so I just keep learning all this interesting stuff about CF. The last two studies I was in were about gene-based research. The ProQR study was a proof of concept study that introduced RNA into the nose. But the results of this study did not Oh, no, the results of this study did support the potential for meaningful therapy. So the proof of concept went forward. I was also in the Translate Bio study in which I inhaled mRNA directly into my lungs. And those were weekly visits with a lot of observation time before I could leave the clinic. For me, that it seems the study did not result in the improvement they were looking for. My FEV went up, but I was also on the placebo. So there's that. But participating in clinical research study really does give me an opportunity to learn more about CF, but also to experience new and promising treatments. But furthermore, what I really like about participating study is that I get to give back to the community that I have benefited from in so many ways. So thank you all for doing the studies and to all of our clinical staff who make them possible for us. Thanks.
Thank you so much, Laura, for sharing that incredibly helpful firsthand account. So now we're going to turn to the questions that we received from the CF community. We received hundreds of pre-submitted questions, so we're going to do our very best to get to as many as possible tonight. I also want to make a note that we have grouped them together to try to hit on them most efficiently. So if you don't hear your exact question or you hear it worded a little bit differently, please know that that's just us trying to get to as many as possible and hit multiple ones where we can. So if we're not able to get to it in the pre-submitted question section that we're answering right now, hopefully it will be included in the Q&A section. And then there's always the blog post recap that's coming after this, where we're going to try to hit on those that we can't get to at all tonight. And so panelists, we're going to start off with the questions for you. And our first question tonight is, and some of these questions, I should say, will hit on things that have already been discussed, but I know, at least for me, it's a lot of information, it's complicated information, so sometimes it's really helpful to hear it in different ways, from different angles, and just multiple times. So some of these will may ask things that you've hit on before, but just sort of asking it in a different way. So the first question is, will gene-based therapies work for all mutations?
Yeah, I can take that one. So yes, so the idea is that you're going to replace with either mRNA or gene therapy, you're going to replace the problem. So with ASOs, we talked about those little tiny messages, that would be for specific mutations, but for gene therapy and mRNA therapy, those would be for any mutation.
And I know you covered some of this before, and this may be a question again for you, Jen, but if anyone else wants to chime in. So you talked a little bit about what could go wrong, from a clinical standpoint, but from an actual patient perspective, can you talk a little bit about the potential side effects and what could go wrong, like in my body, if I'm on one of these therapies? I'd love to hear your thoughts on that.
Well, I would say, first, as Ahmet sort of alluded to, we do test these in animal models oftentimes beforehand. So for example, a couple of the therapies had tested it in monkeys or primates who are very close to us in terms of what their lungs are like. So we do that before we go into people with most of these therapies. But so one of the things I mentioned is that when you inhale things, your body's natural reaction is to try to get rid of it, either by moving it up the cough ladder or by using your body's defense system to get rid of it. And so one of the possible issues is whether your body's going to try to fight it off and create a lot of inflammation in your lungs. So that's one possible side effect. And if you have inflammation in your lungs, that can lead to coughing and wheezing and shortness of breath. Also, you could, in some of the older trials, people had fevers because, again, your body, if you get a virus, that's what it does, is it tries to fight it off with a fever. So I think those are some of the acute things. When you're inhaling something new, there's some people who just don't do well with inhaling liquids. And so right now, all of these therapies, probably in the beginning, that's how you're going to have to take them. So if you're somebody who has a lot of trouble with even simple things like hypertonic and Dornish, you might have some trouble, again, feeling short of breath or coughing with these kind of therapies. Over the long term, like Marty, I don't know if you wanted to jump in. I saw you were off. I'm like, I could keep talking for the rest of the hour, but if you wanted to jump in, go ahead.
No, just agree totally. I think those are, unfortunately, there's a little bit of an unknown. We have some history with these. We hope that we're designing that the community is designing safer therapies up front, but there is a bit of an unknown. And so that's the reason there's a lot of safety valves put in place to try to minimize the risk. And certainly we recognize a safety signal to prevent risk to others before it, becomes too much. So I think it's part of the trust that has to happen with, especially with these types of therapies with the community, that those safeguards are in place.
And I just add that, Ahmet also sort of mentioned there are multiple layers of people looking at things, but this gene therapy trials are also built on the backbone of decades of trials experience in the CF community. Some of these questions were probably asked when ivacaftor was first done in a phase 1 trial. No one had known what these drugs may or may not do in the body, but we had to have small trials, so-called phase 1 trials, to start identifying what potential concerns might there be. And the FDA, there are critics who may say the process is a little bit slow, but at the same time, it's really geared towards initially looking at safety signals. And so these phase 1 trials tend to be very small, always include adults only. They rarely, if ever, go to children first. And they also are really geared towards looking at short-term safety. And so it's not about, someone being on a therapy for a long period of time, but really being on a therapy for a very short period of time, which gives you some answers. And then it's sort of a builds to the next phase and the next phase and the next phase. And I mean, we're hopeful to accelerate that pathway, but we know that of the large group of studies that Jen showed in her slide, some will make it past phase 1 trials and probably some will not. And that was the case for modulator trials. That's been the case for anti-inflammatory trials and and I think the CF community is familiar with that. As much as we would love to move things as fast as we can, having all those safeguards is also equally important.
And I would just add one more thing to what Jen was talking about, the appropriate studies preclinically before they get to humans. When the TDN and the gene therapy work group, when they're giving feedback to sponsors, it is often that they need to go back to the drawing board and do more of those studies with the appropriate, animal studies or appropriate cells to make sure that what they're suggesting is actually working, and that we know that they're being done in a model that is similar to humans. So that type of feedback is often given. And, I also alluded to that there are other, chronic illnesses where gene therapy is being utilized, and we're learning from them as well. And the different therapies that we're looking at are enhancing, because the gene therapy community is a close one, and they learn from each other. And so, some of the things that we've seen in the past are hopefully being avoided in the future.
Yeah, and the last thing I would just add is that we talked about consent and how important consent is, and that's really true for these therapies, because the safety risks are going to be slightly different depending on what vector, like what way you're using to get the material into the cell. So whether it's a lipid nanoparticle, and even the different viruses that we use have a little bit different risks. And so it's really, really important to go through and ask all the questions you need to about the safety of a particular vector or carrying capacity into the cells. That's really, really important.
And Laura, you mentioned the sort of ability to travel long distances for trials. And I think in these particular trials in specific, there are only going to be a few centers doing phase 1 trials. And the physicians there, the site investigators, are working very closely with the sponsors and CF Foundation or other sponsors to learn really about the safety of these particular therapies that maybe your home institution may not know about. And so, the ability to travel distances is important, because we're going to need to find people, as Jen said, who may not have participated in trials before, or who may have been nervous to participate in trials, or may not have been eligible to participate in other trials. But we also want to support them to potentially come far from the comfort of their home center. And we will have to work closely with their care center, the clinicians they trust the most, but also and gain that trust, from a family or a set of people that we don't necessarily know clinically. And I think that's something that the community has really embraced in terms of knowledge and making sure that we all are in this together.
Right. And I just want to say a quick plug here for the emilys-entourage patient registry, because that is a way, especially with these trials that are not going to be at all centers, but if you are a person with at least one nonsense mutation, or really any person in the outlying 10%, I encourage you to sign up for the registry so you can hear about trials directly, as well as through your clinic, and make informed choices about what you want to learn more about and what you want to participate in. It's really a way to equip yourself with as much information as possible. So moving on to the next question here. So you alluded to this in the presentation, but I'm wondering if you can talk a little bit more, or maybe more than alluded to, but talk a little bit more about redosing and what kind of issues can be anticipated or you'd worry about with sort of redosing.
Yeah, so the biggest issue with redosing is whether or not, one, you already have antibodies. So for example, adeno-associated virus is like out in the world. And so there are some people who are going to have antibodies already for that. And then also some people will get the gene therapy dose and then develop antibodies to it. So the question is, can we redose you, or will you, have a reaction because you already have the antibodies? And so there are ways to prevent the antibodies, and data I can say because I presented at the European CF conference is that in one trial we're using prednisone to prevent a reaction, and in some people who already had antibodies that pre-existed, they actually still did okay with the therapy, fine. But there are also ways now, because our technology is a lot better than it was 20 years ago, that we may be able to change it just a little bit enough so that you wouldn't have a reaction. So one of the things that I didn't get into in detail is that when a virus goes into your body, there's something on your cell that the virus binds to. It's like a little lock and key mechanism, basically. And so if you change that just a little bit, so it still fits into the lock, but not so much the body's like, oh, hey, that's that. Let's attack it right now. It might be enough to get around that problem.
Marty?
I agree with Jen. I think there are some unknowns. One safety valve that we haven't gotten into very much, this is more of a clinical discussion, is that I have a little bit of experience with from the laboratory side, is a lot of this testing is done in advanced animal models. So when you have a significant potential safety concern or risk that we can't predict, even extra level of testing. And I can tell you that the CF research community is spending a lot of effort making sure that these are tested in lots of animal models, not just they're going to work, but also the safety, because there's a lot we can predict from that. And in fact, I can tell you there have been gene therapies that have been stopped at the level of the animal model because there was even a inkling of concern at that point. So there really is a lot of effort put into making sure the safety is there, not just trying to get products that have a good effect, but also are very safe, at least the best we can tell.
Okay, great. So one question that's come up from a lot of people is, will gene-based therapies work for people with CF who have had lung or other organ transplants?
I think the long and short of that is until we're out of the lung delivered therapy era of gene-based therapies, the answer is no. Second of all, the safety concern of someone that's got a transplanted organ because of infection risk, altered immune system, those are going to be they're going to be excluded from these initial studies. Now, in the future, that may be the case, but I think for the foreseeable future, that will be an exclusion from the studies if you've had especially a lung transplant or other organ transplantation. I think you can probably see that for quite some time.
Because your lungs, the new lungs already have the correct CFTR.
Right. There's also the difficulty of efficacy, but even I think a systemic therapy would probably be excluded because of the immune issues and whatnot. So,
Ahmet? Oh, I think you're on mute.
There we go. Hello. There's also, other complications with lung transplant. We just spoke about this at a different conference about, cancer risk. And if we're trying to look at other, outcomes like not increasing risk of cancer with, gene therapies, then we want to start with groups where that is minimal, and then obviously the lung delivery. But also because I stumbled on this earlier, I did want to just throw another plug to Emilys-Entourage registry and the matchmaking program, because that is how, for a different study looking at a different way of helping people with stop codon mutations, we really found people to join our clinical trials. So definitely register for those who haven't, because it's really important to get these studies done.
Thank you so much for that. So I'm just wondering, and I can already imagine what some of the answers to this will be, but for the CF community, our benchmark is really modulators and how those impacted people. So I'm wondering, and I know there's all different types of gene-based therapies, but what kind of impact do you expect gene-based therapies could have for people with CF compared to modulators?
Emily, I think that's a great question. I think, we think the impact of modulators is because CFTR function is restored. And so, I think that the very basic level, if we can restore CFTR function in the lungs and the airways and other parts of the body, we hope that we would see similar clinical effects, if not, even more. But I think it's really early to know from a clinical perspective. And Jen mentioned the difficulty around sort of what outcomes are we really measuring, that we are able to measure. Lung function clearly will be a part of it, but it probably won't be the only thing. But I think that's at least the hope and the goal.
Yeah, it is a really great question, and I wish I had a crystal ball to answer it, because again, it's going to be variable, probably based on what vector or whether it's the lipid nanoparticles that you're using, but also we just don't know what percentage of cells have to be corrected to get a specific amount of response. And in my mind, I was one of the people who was really, I'll just say, critical about one of the drugs that was more modestly effective. I was like, really, are we going to approve this drug? But then as we saw the data, it showed that, again, as I mentioned, people were stabilized. And if we can stabilize people until they get to the next better therapy, that's a huge win. So in my mind, and others can disagree or agree, but if we could even get a little bit of improvement in lung function, and stabilize, and people have less exacerbations and they feel better, that would be a huge win for people who have no therapy right now. Ultimately, we want people to have, Trikafta effects everywhere in their body. But even if we could get a little bit of that in the beginning, I think that would be fantastic.
I agree. I think we just, we haven't really finished in the current area, haven't really finished one of these trials yet to know what we're going to be able to know. And so part of it is we've got to make our way through a few of these, just like the early modulators, getting through a few of them to know what the benchmarks look like to better design the studies for future. It's just a reality of doing these kind of studies this early, phase we're in.
So just sort of following on from that, for people who have mild, moderate, advanced, or end-stage disease, can you speak to sort of the potential benefit of gene-based therapies for those different stages of lung disease?
Yeah, I think that's another really great question. One of the things I'll point out is that, again, because we are still learning about the safety of these therapies, people with really advanced disease are going to be excluded at first because we don't want to cause people harm as we try to figure out whether or not these work. One of the things that we've seen with the modulator studies is that even in people with really severe disease, we can make improvements. We can't reverse it back to when they had mild disease, but we can make some difference. And so we don't, again, it's a crystal ball sort of answer. We don't know the answer to that yet, but in the beginning, we'll exclude people who have really severe disease. And if we can prove that something's safe, I think that people who have more moderate disease and maybe even severe disease could benefit, although they may not get the same kind of improvement just because we can't reverse some of the scarring like we saw in modulator therapy.
I think we do want to have a broad base of individuals participate in trials because we learn from each individual who participates in a trial, even in these small trials. And I think it's important for us to understand where these differences are early on. I also think, the reasons why people join trials, and I think that there's been some thought about that in the CF community, but more broadly, like, why should you join a trial? Is it for yourself? Is it for the community? It's probably both. And most people with CF, and Laura, you may disagree, but I think most people do it not just for themselves in terms of a benefit, but really because they feel a part of the mission and want to help advance science and advance sort of the field forward. And without that, real active participation from people like yourselves, none of this would get done.
Yeah, Laura, maybe you can say just a couple words about why you chose to participate in the trials.
Well, I'm always eager to find out about a trial, and I like to participate because I actually enjoy the experience, even if it's cumbersome and time consuming. I do, I can try new therapies. I got to experience that little compressor with the Translate Bio study, and so there's some benefits to me, but I have a lot of gratitude to people who did these studies a long time ago, so that I have, when I'm on board, right when I started, I had Pulmozyme and Toby available to me. So I think this is a great community for people coming forward to help themselves and help others and work together with the scientific community. I think I don't know,
There's a lot of altruism.
it's a good club to be part of, in my perspective.
Yeah, that's great. Yeah, we're working on the backs of a lot of people that put in a lot, and also there's a lot of altruism in the CF community, which is pretty amazing. And actually, one other question for you, Lars. I'm wondering if you can speak a little bit about how the experience of participating in the ProQR and the Translate Bio studies, how did that differ from other clinical trials, non-gene-based therapy trials that you participated in?
Both of them had some observation time after the dosing, so that was new, and more frequent visits, I recall, than some of the earlier studies, either for dosing or for observation. So it's time consuming, but it was also, we use the negative pressure rooms for these particular studies, and I don't know, it seems like maybe it's time, but it's all on a new horizon too, so I get to be part of that. And I think every different study is a part of a different approach or a different direction that we're going in.
And Laura, I think you mentioned earlier that you had some outcome measures that you hadn't had ever in the clinic. So like the nasal potential difference measure and lung clearance index.
Yeah, I'd never done those tests before. So I ask a lot of questions. I'm pretty curious. And we did have somebody who was really excited about administering the nasal potential MPD and potential differential. But so I got to learn about those kind of tests that were part of earlier CF care. I think there were ways, but we're still using them also.
Thank you. So I want to ask, for people who are on modulators, do they stand to benefit from gene-based therapy too?
Yeah, that's a really great question, Emily. And the answer is probably yes. But in the early studies, we are generally excluding people who are on modulators because we need to be able to see if the drug by itself is making any difference. Because obviously, as I mentioned, there are people who don't tolerate modulators. And also, it's important to keep in mind that when we report the data from trials, we're reporting the average response. So some person might have responded with their lung function, that's the average, say 15%, but other people may have responded with a lung function improvement of 30, and some may have had no lung function improvement at all, but maybe they have less exacerbations. And those folks probably do want to participate in another trial where they could actually see some improvement in their lung function and not just stabilization. So I think ultimately the answer is probably yes, but in these early trials, we'll exclude people because we won't be able to tell the difference between what's working and what's not.
Makes sense. So, another thing that's very much in my mind is that there's going to be a lot of multiple trials in the same therapeutic area coming up, and we're lucky to have these different options. But how should people go about choosing between the options?
That's another really excellent question. I think that what's going to happen is review with your physician and with the primary investigator so that you really understand what the risks and benefits are and how your schedule is going to work with the trial demands, because some of them are more demanding than others. So I think all of those things are going to have to be a factor. And it may be because all of these studies, if there's travel involved, the company will pay for you to get to the site. But can you miss that much work, and how far is it? Like, do you have to fly to New York from Colorado, or can you fly down from Montana, which is a little less far? I think those factors will play a role.
That's great. And the final question of the pre-submitted questions before we turn to the open is, a big question that I think is on the minds of many people with CF, which is, what is a realistic timeline for these gene-based therapies? So how close are we to seeing these as viable treatment options, and for both adults as well as for kids?
I think we're in early phase of a couple of therapeutics and some others in the same bent. So I think realistically, let's say that the ones that are currently in what may be called phase 1, early phase 2, make it through that phase, that they've got some time on those, probably another 6 months to a year to finish those studies. And let's say it marches through, probably looking at still 2 to 3 years from getting there with the first of these therapeutic rounds. Maybe quicker than that, but that's being realistic with it, given that the processes have been a little slower than we hoped, and enrollment's challenging. And then so others are behind that. So I think realistically, we're a couple of years off from the first of these therapeutics. I think is just honest. Others have any other insight on that, but I think that's probably the most optimistic we could be about it.
I would agree, Marty, but I think, if anything has taught us, the trials for the modulators, I think, surprised me as a clinician about how rapidly they really progressed and developed. And once they did, how rapidly they progressed into pediatric and younger kids, So
yeah, absolutely.
It is, I think the beginning is a jog and a marathon. But I think once things start showing some efficacy, and we hope that some of these therapies will, that the pace will accelerate. And ultimately the goal is to treat kids. I think that's, as a pediatrician, that's obviously something we talk to our families all the time, is that we would like to initiate CFTR altering therapies as early as we can, because we think that will prevent later complications, or at least delay them for a significant amount of time. But we also have to be very careful about trials in kids. And so, I think there's a balance, but I think the realistic timeline that Marty laid out is I think fair.
And I'm just going to give a succinct answer. I agree with everything that was just said, and I am definitely optimistic. We've seen so many great wins, but also I've learned to not promise and not to, make any predictions. And so we have to just go through these steps one day at a time, but I am hopeful that we'll get there.
And I'll just add a quick answer as well, in that we have to also follow what the FDA wants us to do, because these therapies are new, and the follow-up for them that the FDA is requiring is a little bit longer, again, depending on which vector or carrying vehicle you're using. The follow-up is longer, and so we have to see that long-term follow-up too before we start dosing a lot of people with a new therapy.
And I'll just say one final word on that before we move to the live questions, is that a big barrier for those trials happening, turning into real treatments, is recruitment for trials. And so the faster that we can self-organize as a community, and the faster that we can recruit for trials in really informed and thoughtful ways, the faster we can get those treatments, approved and in the bodies of people. And so that's one of the goals of our clinical trial matchmaking program, is to help matchmake as fast as we can, and as well, and and empowering the CF community always with the information to make the right choices for themselves. And so with that, I'm going to shift to our live Q&A session. And so the first question tonight is are there ways that we can make gene therapy trials less demanding in terms of the time required for participants?
The short answer is no, not for the phase 1 trials, because again, we have to be really, really careful that we're being vigilant about making sure that no one is harmed as we start these really new therapies. Later on, when they're in later phases, they'll be less onerous, but in the very beginning, we have to be incredibly careful.
And your CF community member who is looking at those protocols is always pushing back. And so we just have to, make sure that we're at a good balance.
Thank you. So next question is, what types of outcomes and measurements will be examined to determine efficacy in these trials?
I'll take that one. I think there's two types of outcome measures that we look at. One is safety, and to some extent, some of the things that we look at for efficacy, which is effect, I'll discuss in a second, but are also safety measures. But the most important early is safety endpoints like blood work, lung function testing, which is an efficacy endpoint in later studies, is a safety measurement up front, symptoms, that kind of thing, antibodies being measured in blood, et cetera. Then there's efficacy endpoints, and those come in two flavors in CFTR activating therapies. One is the CFTR protein working and turned on, and that looks like nasal potential difference, maybe sweat chloride if it was a systemic therapy, lower airway potential difference, those kind of things, maybe biopsies looking at that kind of stuff, which is being done in current. And the other is clinical efficacy. Like, does it make you feel better? Does your lung function improve? Do your clinical symptoms get better? That kind of thing. Do you gain weight? Things that we might expect, like in a CFTR modulator therapy. So the menu is similar, maybe a little bit broadened because a few extra options of the menu because of the unique nature of these studies, but a similar menu to modulators, especially in the early phase studies.
One thing I should point out, sorry, I know we're supposed to hurry up, is that for these studies, unlike the modulator studies, many people will be asked about bronchoscopy. So going down into your lungs with a camera and taking little pieces of tissue to see if we actually got the gene where it's supposed to be. And that's something very different than with modulator trials.
Good point. Good point.
That's great. So how might inform, you were talking a little bit about informed consent before, but how could informed consent documents be constructed if risks and side effects are unknown in humans? So can you talk a little bit about those ethical considerations?
That's a great question. And I think, this is one around clinical research and human subjects research that this is why IRBs are in existence. This is why sponsors work with the community to come up with language that is both accurate, but also expansive, but also leads to some unknowns and is quite broad. But it's also why you spend a lot of time as a trial participant meeting with the study team prior to enrolling or agreeing to enroll. The documents themselves often have stock language because they also serve as legal documents. And the FDA has some language that they want us to have. Institutions have language that they want us to have. And for gene therapy trials in particular, many institutions, academic institutions, have developed their own gene therapy working groups, talking about lots of different therapeutic areas that want to think about different things. And so, as Ahmet mentioned, all those layers go into sort of all the wording and phraseology that goes into those documents. And they can often be 20, 30 pages long. And that's why it's so important to have that conversation with the study team and really go through it in excruciating detail, because they are sometimes very specific and sometimes intentionally broad.
And I'll add that one of the purposes of tonight's town hall is to, provide basic information and sort of inform, equip all of you with the tools to know the questions to ask. We can't possibly get to everything, but to really make you feel empowered and equipped to ask the right questions and consider the right things and, reach out to the right people when you're considering those trials. And so next question here is, are people outside of the US able to participate in trials?
It largely depends on the sponsor and where, but these trials will be conducted, some of them outside the US sphere anyway. So I think it depends on where you're coming from and what the individual sponsor has agreed upon as far as international travel. So I think it's study dependent, but many studies that, especially for rare treatments like this, there have been opportunities for outside the US to be at least considered.
And we've looked into this, and there are clinical trials going on elsewhere, not just in US and Canada, but coming here, that there are some complications, including having health insurance, and that can sometimes be an issue, but you can purchase that. And there is actually nothing against someone coming from another country. There's just some barriers that just need to be jumped.
Logistic frameworks mostly, yeah.
I also want to mention that on our registry, one of the things that we do is we look at the data of where people are located, and we work with companies to inform their clinical trial sites. So if we can show that there are large groups of people in particular countries or other areas, we advocate for companies to set up trials in those areas. And so, not trying to plug the registry again here, but that's another reason to sign up, is because we can help push to get trials in those locations all around the world. And so next question I want to ask is, you talked about this a little bit, but wondering if you can hit on it again. Where can people get more information about the gene therapy trials that are available now?
So yeah, I mentioned, and people will be able to see the specific websites when they look at the video, but you can go to clinicaltrials.gov and search cystic fibrosis and gene therapy or mRNA, and it'll bring up all the trials that are happening right now or the ones that are getting ready to start. And you'll see some of the older ones that have already completed, but you can look up details like how often do I have to come and what are the basic criteria to get in and where is it being conducted on clinicaltrials.gov. Also, if you look at the sponsors website, they sometimes have descriptions that are meant for people who aren't necessarily in science to read about what their drug does or what they think that it does. Obviously, they can look on your website. There are other websites where they give some more layman-friendly language in terms of what's going on right now and what's coming up.
Right. And then wondering if maybe we can just go around really, really quickly, make this really brief, but I'm wondering if you can all share like what, in this new gene-based therapy era, it really feels like a new era for the CF community. I'm wondering if you can just share in like one word or just a couple words, like what makes you most excited, as people with CF and clinicians treating CF right now?
I think I'm most excited about the fact that there are so many companies that are jumping in and trying to create drugs for this really relatively rare disease. So the more shots on goal that we have, the more likely it is that we're going to find a therapy for everyone. And so it's really exciting to see so many people who are so interested in trying to find a cure for CF.
I was going to use a word of momentum. I think that's kind of what Jen's getting at, is there seems to be a head of steam and a momentum about this happening now that was kind of not there even a couple of years ago. And so I think that's will get us there, because there's a lot of momentum from the community and the research and everything.
I would just add that there's incredible brain power, incredible people who are interested in trying to find a cure for cystic fibrosis. And there's that altruism that Emily mentioned amongst the providers, caregivers, and people in the CF community. And I think that is, just always incredible to me. And to be able to, in my career, hopefully see the whole spectrum of what's going to happen as we look at how we manage CF change. And then really quickly, I'd also throw a plug in for Marty and Deepika Polanini, who have an introduction to gene therapy YouTube video that you could search for as well.
Well, and I'll just sort of say all the above, but I really think it's exciting and so impressive that we as a community really have committed to finding therapies for everybody with CF. And, we didn't just say, wow, we have some great therapies for some people, but we're really emphasizing everybody, the marginalized, the people that are not able to access therapies or may not tolerate therapies, or those who simply are born with mutations that may not be amenable to these therapies. And I think, there are communities where that wasn't necessarily a given that it would happen. And I think that's what excites me and really empowers, I think, all of us to sort of continue on this work.
And I'd like to offer that going forward, it's all very exciting, but I am so comforted by all of the layers of safety and scrutiny that the therapies are receiving, and for the diligence and enthusiasm of everyone who is working towards making our lives better for a very long time. So thank you all.
That's perfect. I think that's a great way to wrap up our questions. And so I want to thank all of our incredible speakers for joining us tonight and for these really thoughtful presentations and answers. And I also want to thank everyone that submitted and asked questions live tonight. These have been fabulous questions, and it's really been a productive and a very hopeful conversation to me, and I hope for all of you. I want to also say a very heartfelt thank you to the emilys-entourage staff that made tonight possible, and especially to Beth Lavin, who's behind the scenes, without whom this event would not have happened without her leadership. So thank you, everyone that made this a success. This is an incredibly exciting new era of gene-based therapy for the whole community, and especially for those in the final 10%. Finally, we have some potential options in the pipeline. And, this has been a long time coming. And so it really, if I was going to add my one word, it would be finally. And so I say this with a healthy caution always, because, but there's really never been a more hopeful time than there is today for every single person in the CF community. And that's thanks to the incredibly hard, persistent, and devoted work of tons of people, clinicians, companies, and regulators, but and most, and always closest to my heart, the actual CF community, people with CF and their families, that blood, sweat, and tears goes into all of this. And so what I'm most excited about is that there are multiple trials in multiple therapeutic areas being led by multiple companies. And so really a diversified portfolio, and hopefully multiple will work. And that's really exciting to me. And so, our sincere hope is that our community can be and really feel equipped to make smart, informed, and thoughtful choices as you all consider these different trial opportunities. And so I want to share just a few final reminders. The first is, I want to ask you to look for that post-event survey at the end of this webinar. It's just 4 questions. It'll take you a matter of seconds, but that feedback is so helpful for us. The second thing is, we are going to be coming out with a blog post recap addressing questions we couldn't hit on. And the best way to ensure that you see that is to join our newsletter list and follow emilys-entourage on social media. And so I think you should be seeing those on your screen, but you can also always go to our website and get all those links and follow us from there. We've mentioned it a few times, but our patient registry is for anyone with at least one nonsense mutation of CF, and hopefully in the near future, we'll be expanding beyond that to those in the 10%. So please consider signing up for that. It's the best way to hear about trials that you may be eligible for quickly. And also we would love for your help spreading the word, because as we said, the more people and that we get on there, and the more parts of the world that we have represented, the more we can really advocate to set up trials that reach everyone with CF. And then finally, our biggest event at emilys-entourage is an annual gala. It's coming up on October 14th. We have an option to participate virtually. It's totally free, but you do have to register in advance. And so we would be so honored to have you all join us if you're interested. And you can find more details about that on our website. And so with that, I want to just thank our speakers again. Thank you to our question submitters, our staff, and everyone who joined us tonight and made this such a wonderful and informative discussion. Thank you and good night.